Alright, to start protein synthesis part 2, we're going to quick do an overview of transcription. So remember from the last video, DNA is what holds all of our information. It's in the nucleus, but it can't leave the nucleus. To start transcription, the DNA unzips and a complementary mRNA strand attaches to it. Once it is complete, the mRNA detaches from the DNA, leaves the nucleus, and ultimately it travels in the cytoplasm toward the ribosome. Now, before we get started on translation, I want to go over some vocabulary, some words that are going to be used that I want to make sure we're all on the same page with. So when we talk about genetic code, we're just talking about all the instructions to make proteins. A codon is a set of 3 nucleotides on the mRNA. So this points it out really well. So on the right, the U, C, and A you see right in the beginning of mRNA, those 3 together would make a codon. Now an anticodon is the 3 nucleotides that are attached to the tRNA that are complementary to the codon on the mRNA. And so we'll talk about that more later in their interaction. And the last one is just amino acid. So recall, that's our monomer for proteins. It's the building block, and it's brought to the mRNA by the tRNA. And the amino acids are held together by peptide bonds. So our purpose for translation is to take this mRNA that's left the nucleus, it interacts with the ribosome to ultimately make a protein. So all of this is taking place in the cytoplasm and at the ribosomal site. So again, we start with our mRNA and we end, our final goal for the whole central dogma is the creation of a protein. So when we have our mRNA, we ultimately have a number of tRNA that are going to be bringing over amino acids. But the whole point of this is that the mRNA message that's coming from the DNA is just being translated into a protein. That's where the name actually comes from. It's translating a message. So the beginning steps of translation obviously take place at the final stages of transcription. So that mRNA that is coming from the nucleus, traveling through the cytoplasm, attaches to the ribosome. And you can see this below, where the large subunit up top is blue and the smaller subunit of the ribosome is brown below. So that mRNA attaches to the ribosome. The ribosome reads the mRNA codon, remember the 3 nucleotide sets on the mRNA, like a triplet. That's another name for it, a triplet. And the tRNA, which you can see, it kind of looks like a hairpin, comes down. It's kind of like a taxi. It picks up and drops off amino acids that match up with the mRNA codons. So as you can see, there's a tRNA coming down on the left that has UAC, and that's going to match up with AUG down on the bottom. So our 4th and 5th steps are tRNA continues to drop off amino acids. Proteins are made up of many amino acids. And they can form really long strands. So the ribosome binds the amino acids together with something called a peptide bond. Now, there's a stop codon that ultimately is just kind of your stop sign. It says, okay, this is the end of our protein. We do not need any more amino acids. And when that stop codon is reached on the mRNA, the ribosome releases the completed protein from the tRNA. Now, there are 20 different amino acids that can be created. So if you look here, there are a number of different combinations that you can see. And so we'll learn more about this wheel later. But the different combinations, each combination of letters leads to a different type of amino acid. So I can't do the math in my head, but as you can tell, when a protein's being created, there are an infinite number of possibilities for amino acid sequences, protein sizes and complexities. But ultimately, it all boils down to the combination of codons on the mRNA. All right. Similar to the last video, I want to see if we can take a DNA sequence and go all the way from DNA to a protein. Okay, so we're going to go through transcription, we're going to go through translation. So here's our DNA sequence. Go ahead and try and get our mRNA sequence from this one. Hopefully it looks something like this. Now, from here, we have to create our codons. Remember, codons are triplets. They're sets of 3 nucleotides along the mRNA. So see if you can separate that mRNA sequence into codons. All right, here's what we should get from that. So we have 5 different codons here. Now, see if you can use the wheel in your notes to come up with our amino acids. See if you can use that wheel, find the codon sequence, and translate them into amino acids. All right, here's what we should get. Again, see that the stop codon is at the very end. That's the end of our protein. Okay, let's see if we can go through all of this. Let's see if we can go through a summary of the central dogma. So, at the very beginning of transcription, DNA unzips and the mRNA attaches, all right, a complementary mRNA strand attaches to the DNA. Once the mRNA strand is complete, it detaches, leaves the nucleus, and travels into the cytoplasm toward the ribosome. Now is where translation comes in. At the ribosome, the tRNA attaches to the mRNA and brings amino acids. This continues, and the amino acids are bonded with peptide bonds until the stop codon is reached and the protein is released.